Neurons have three parts, a cell body, and two processes, dendrites and an axon. Dendrites are the smaller and the main receptive processes that receive signals from other neurons and convey them to the cell body and then to the axon. Axon is the longer process and it carries the information away from the cell body in the form of bioelectric signals called nerve impulses. The end of the axon is tipped by small swelling called a synaptic knob, and it makes a synapse to either the cell body or dendrites of another neuron. Longer neurons are covered with fatty, protein lipid, whitish, and segmented myelin sheath. It protects and electrically insulates the neuron as well as increases the transmission speed of the nerve impulse in myelinated neurons. Potassium is the main intracellular positive ion and sodium is the main extracellular positive ion. Distributions of these ions are maintained by sodium-potassium pump. At resting conditions, the cell membrane is less permeable to these ions but more permeable to potassium than sodium ions. The cytoplasm of neurons contains impermeable negative ions such as phosphate, sulfate, and negatively charged proteins. The distribution of ions creates charge differences, positive at outside and negative charge at inside of the membrane. At resting conditions, membranes are polarized and the potential difference is called resting potential, which has a value of -70 millivolts. Neurons are excitable, some of them sense temperature, pressure, or light outside the body. Such stimulus locally affects the membrane potential, which originates the nerve impulse at the initial segment of the axon, known as the trigger zone. Other neurons respond to signals from inside of the body, coming from other neurons. Presynaptic neuron brings the impulse, signal, to synapse, which stimulates or inhibits the postsynaptic neuron or any organ. When a nerve impulse reaches the axon terminal, voltage-gated calcium channels open, which results in the inward diffusion from extracellular space of calcium ions. An increase in the calcium ion concentration in the synaptic knob results in the fusion of the synaptic vesicle containing neurotransmitter to the membrane and the release of the neurotransmitter through exocytosis. The binding of neurotransmitters to the receptor on the postsynaptic neuron results in the opening of the chemically gated sodium channel and inward diffusion of sodium ions into the cell. The inside of the membrane becomes less negatively charged, which results in depolarization of the membrane. The multiple stimulation by presynaptic neurons results in depolarization to the threshold level, which is called threshold potential, which has a value of -55 millivolts. Threshold potential results in the opening of voltage-gated sodium channels present in the trigger zone of the axon and inward diffusion of the sodium ions from extracellular space. The left side of the figure shows the trigger zone of the axon where action potentials are generated. Stimulus below the threshold level is called graded potential. At resting conditions, voltage-gated sodium channels are closed and the membrane is polarized, having a membrane potential of about -70 millivolts. Any stimulus or signal that results in the threshold potential opens the channels and increases the sodium permeability. This leads to positive on the inside and negative on the outside, which results in action potential. At the peak of the action potential, membrane potential reaches plus 30 millivolts. Soon after the peak of an action potential, voltage-gated sodium channels get closed and voltage-gated potassium channels open, increasing the potassium permeability toward extracellular spaces. As the potassium moves out, the inside of the membrane gets negative once again, thus repolarized. The left side of the figure is showing the propagation of an action potential. At the site where the action potential is generated, positive ions on both sides of the membrane move toward more negatively charged ions, which create a local current. This current depolarizes the neighboring area of the membrane in the forward direction, which opens the voltage-gated sodium channels and generates the action potential there. At the same time, the backward side of the membrane is under repolarization to achieve the resting potential. The area which has just generated the action potential, the sodium channels in that area get inactivated, so no new action potential can be generated here. In this way, the action potential travels in the forward direction toward synaptic knob. The propagation of action potential in the neuron is called a nerve impulse. An unmyelinated axon conducts action potential over its entire length. In myelinated neurons, the threshold results in the action potential at the trigger zone. It produces the local current which travels away from the trigger zone through the cytoplasm of the axon. As this local current reaches the first node, it excites the membrane to its threshold level and generates action potential there, as well as sends the electric current to the next node. In this way, the action potential in myelinated neurons jumps from node to node. This increases the transmission speed of nerve impulses in myelinated neurons, approximately 120 meters per second, whereas in unmyelinated, 0.5 meters per second. Let's summarize the nerve impulse conduction in the main 6 steps. At first, action potential or nerve impulse arrives at the axon terminal of the presynaptic neuron. Then second, voltage-gated calcium channels in the axon terminal open, and the concentration of calcium ions in the cell increases. Third, an increase in the calcium ion concentration causes the fusion of the synaptic vesicles to the membrane and the release of the neurotransmitter in the synaptic cleft through exocytosis. Fourth, the neurotransmitter binds to the receptor and opens the chemically gated sodium channel on the postsynaptic membrane. Sodium entry into the cell causes membrane depolarization. Such depolarization is locally or graded. Sufficient stimulus from the presynaptic membrane depolarizes to the threshold level. Fifth, as the threshold potential is achieved, a voltage-gated sodium channel opens in the trigger zone of the axon, and sodium concentration inside the cell increases, generating the action potential. Sixth, action potential propagates with the help of the movement of ions and local current, which is called a nerve impulse. Finally, the nerve impulse reaches the axon terminal, and in the same way, the impulse will be transferred to the next neuron.